Well, good afternoon, good morning, good evening, wherever you are. Welcome to MycoTalks, which is coming to you, well, at least from around the world in 3 different countries, but we're based at the University of Exeter. And as you'll see from that first slide, we've got 2 very exciting speakers for you today, Iuliana Ene from the Institut Pasteur, and then Bernhard Hube at the Leibniz HKI in Jena in Germany. But before I introduce the first speaker, Jules, today, a couple of other things just to remind you about the MycoTalks series. The idea of this is to share our expertise around the world, taking note of diversity, career stage, gender, geography, etc., to give you a wide diversity of types of talks. We always have 2 talks on any aspect of medical mycology. And we have the 2 talks back to back, and then we don't have any questions between them. We have questions at the end of the session. And at that stage, you'll be thinking of questions for both speakers. So we invite you to send your questions to us, and please do use, importantly, the Q&A button for this. Don't use the chat. We're not going to be monitoring that. And please do indicate, if it's not obvious, to whom the question is addressed. We will address them, myself and my co-host, Sarah Gaffin, will do this, and we will alternate those questions. And if you know somebody who would like to have been here but's missed it, you'll be able to find this MycoTalk on our YouTube channel in due course, so you can revisit it at your own pleasure in your own time. Okay, just to alert you to the one after this series on the 26th of February, another Thursday, we're going to have George Deep and Tamara Doring, so something to look forward to. And then you can see in 2nd of April, Lee and Marty Burke will be following on from them. So a great series to be aware of over the next few months. We also have a sister series, which are called the mycoclinics, which are done every quarter. And this has got more of a clinical field where, as you can see there, the experts in the field present often clinical cases and highlighting challenges and new observations in clinical recognition, diagnosis, management of fungal diseases. So do look out for that as well. And I will now stop sharing and introduce the first of our 2 speakers. So I'm absolutely delighted to welcome Iuliana Ene. And Jules is an assistant professor in the Mycology Department at the Institut Pasteur in Paris, and she established a research group there early in 2021. And she is interested and focuses on the understanding of how fungal pathogens respond and adapt to antifungal drugs, particular emphasis on population heterogeneity, tolerance, heteroresistance, and genome plasticity. So she's combining functional genomics, experimental evolution, and quantitative approaches, and she studies how Candida species survive under drug stress and cause persistent infections. And prior to joining the Institut Pasteur, she was a research assistant of the Molecular Biology and Immunology group in Brown University, and she completed her postdoctoral training with Richard Bennett there. And of course, I personally know her from before that, where she received a PhD from the University of Aberdeen, which she did with Professor Al Brown, which was then part of the Aberdeen Fungal Group, which is now incorporated into the MRC Centre for Medical Mycology. So Jules, you're very welcome. We're very excited to hear about your talk. You're going to tell us about beyond drug resistance, alternative drug response mechanisms in Candida species. So welcome, and over to you.
Thank you, Neil. And while I figure out how to share my screen, okay, does it look good now?
It looks perfect, Jules. Yeah.
Perfect. So hi everyone on all time zones, and thank you, Neil, for the nice introduction. It's really a pleasure to be part of the series again. So we work on Candida species, and we're particularly interested in how they respond to antifungal drugs that we currently use in the clinic. So just as a way of introduction, my group focuses on these species because these are normal human residents, but they can actually cause a lot of infection. So they can disseminate, cause mucosal bloodstream infections, as well as dysbiosis in the gut, and this has been associated with several inflammatory diseases. And altogether, these infections are estimated to lead to approximately 1 million deaths annually after the most recent estimates. And unfortunately, we have no vaccines. We really live in an era of limited anti-infectives. We have very few antifungals, increasing drug resistance and increasing populations at risk. And to add to this, we also have frequent treatment failure, even with susceptible isolates, suggesting that we really don't understand how these cells respond to drugs. So really the focus of our work has been to better understand how cells adapt to antifungals in the absence of canonical drug resistance, and also how can antifungal adaptation be targeted therapeutically. So this is what our main research efforts have been focused on. So in terms of what antifungal drugs do we have, most commonly used antifungals really target a few different components of the cells, mainly the cell wall and the plasma membrane. And also, it's been challenging to develop new antifungals because many of the fungal proteins that are potential targets for therapy are also found in humans with substantial drug toxicity risk. And unfortunately, really, there are drug-resistant Candida strains for all existing antifungals. So we need to do better in this aspect. Among antifungals, azoles are some of the most widely used, both in the clinic and in the environment, and this poses challenges in itself. And this is because these drugs have a broad spectrum of action, they're relatively available, and show less toxicity than most of the other antifungals. And in principle, these are static drugs, so they don't kill the cells, they just inhibit their growth. And the way they work is to target ERG11, which encodes an enzyme that participates in the synthesis of ergosterol in the plasma membrane, thereby destabilizing the membrane and inhibiting the fungal cell growth. So even though azoles have been studied for decades, really for about 40 or 50 years, our understanding of how these cells, how fungal cells respond to them, remains limited. So isolates are typically classified into two bins, either susceptible or resistant, as illustrated here using these disk diffusion assays, really based on their ability to grow in the presence of these drugs. So resistance is often genetic, and it's achieved via changes in drug targets, so by either altering or overexpressing the drug target ERG11, or by changes in efflux capacity by upregulating efflux pumps. However, Candida isolates tend to be actually overclassified as susceptible or resistant based on these two categories. But this does not reflect how diverse the Candida isolates that we recover from the clinic are. So rather than viewing antifungal responses as a binary trait, in our group, we frame it as a continuum of adaptive states. So really what cells do is to exploit diverse mechanisms. So these alternative mechanisms such as tolerance and heteroresistance, and also their population heterogeneity. So the ability of cells to do different things under stress in order to really evade the effects of the drugs and to resume growth under these very challenging conditions. And we think that it's important to study these alternative phenotypes because these are actually very frequently encountered among clinical isolates, and we hypothesize that they could precede true drug resistance. So the two phenomena that are relevant for the talk today are really tolerance and heteroresistance. So I'll start with heteroresistance, which implies the existence of subpopulations of cells that have different drug susceptibilities. However, their resistance levels are transient and cells rapidly revert to susceptibility when the drug pressure is removed. So this phenomenon is transient and really occurs at very low frequencies. So in the lab, we use population analysis profiling assays to quantify it. So we've recently published methods to do this both in liquid and on solid. So essentially this works by counting the number of colonies that grow at very high drug concentrations. So this is well above MIC50 levels. So for example, the heteroresistant isolate is shown here, will show a phenotype that is similar to the curve here, basically a fraction of the population, in this case, a very small fraction, 0.1%, is able to grow at very high concentration, so over 100-fold higher than the MIC of the strain. whereas a susceptible isolate is really unable to do this. So these examples come from Candida albicans strains, but really other Candida species also display this phenomenon, including Candida auris, Lactobacillus glabrata, as well as Tropicalis dubliniensis and others. Probably everywhere we look, isolates will be available to do this to some extent. So similarly, tolerance also reflects a subpopulation phenotype, but in this case, the cells are now more drug resistant. So if you would take some of the cells that grow here in this area of drug inhibition and you redo this assay, you recover a phenotype that is similar to that of the parent cells. So cells are able to grow at slow rates in the presence of very high drug concentrations, so above MIC levels. And this property is stable, is inherited, and is importantly independent of drug concentration. So we know that tolerance is important for clinical purposes because we see an association between high levels of tolerance and the ability of isolates to persist during multiple rounds of therapy. So we think this phenomenon is important to study mechanistically and as it might have implications for management of these fungal infections. So similar to heteroresistance, other species and also other antifungals also exhibit tolerance. So these are some examples of images from Candida auris, Tropicalis, and dubliniensis challenged with an E-strip containing the drug caspofungin, so anakinocandin, and you can also see these population heterogeneity phenomena in these cases as well. So population heterogeneity is really prevalent, as well as tolerance is prevalent in clinical isolates of diverse species. You can see very high levels of tolerance in Candida auris, Guillermondii, dubliniensis, tropicalis with echinocandins. So really this phenomenon is quite frequent and again emphasizes that we really should be looking at it more closely. So in terms of what we know about the mechanism for some of these phenomena, we know from work from Judy Berman and ours that multiple pathways actually impact tolerance. So this includes pathways that control membrane integrity, cell surface, as well as stress responses such as calcineurin, HSP90, TOR, PKC, or PKA. So we were really interested in what other genetic factors are associated with azole responses, but we wanted to do this in sort of an unbiased way. So to do this, we collected a series of clinical isolates, actually not only clinical, but isolates from diverse sources, including environment or from food spoilage. And we decided to phenotype them and see, well, how broad are these different drug responses? How broadly distributed are they? So we measured susceptibility and tolerance using MIC assays by looking at both 24 and 48 hours. And we measured heteroresistance using population analysis profiling. And our assays revealed that this set of isolates actually show a diversity of phenotypes that covers the entire spectrum of drug responses. So we see isolates with both low and high resistance, as well as different levels of tolerance and heteroresistance. So this diversity provided us with a wide enough phenotypic range that is needed for a genome-wide association study. So in addition, we also looked at the distribution of the three phenotypes and asked whether or not they're correlated with each other. So we found that the three phenotypes were widely distributed and only modestly correlated with each other, suggesting largely independent mechanisms between the three traits. So while highly tolerant isolates seem to correlate with high levels of heteroresistance, there are also many susceptible isolates that had high tolerance and high heteroresistance, really pointing to independent mechanism, and that the fact that the relationships between these three phenomena is not straightforward. So our next step was to sequence the isolates, especially for those that had not been sequenced by others, in which case we simply obtained their genomes. And we did the phylogeny analysis to see how widely distributed the three phenotypes were. So this revealed that the isolates both showed a highly wide phylogenetic distribution. So we have isolates from all the major clades, and in fact, we identified several new clades. And in terms of the three drug responses, we found that they cut across all clades, suggesting that our set of isolates is actually suitable for GWAS study. So broad population structure and broad phenotypes. So we went ahead and ran a mixed linear model GWAS and obtained significant associations. This was really our hope to find things that were significantly associated with our three phenotypes. So these three Manhattan plots really showed the results of this analysis. So to summarize, basically we found 57 loci under stringent thresholds. So these were highly significant above these lines. 16 associated with susceptibility, 38 with tolerance, and 3 with heteroresistance. But we decided to also look at the loci that were right underneath this threshold. So we used two thresholds defining both stringent and relaxed loci. So these were in the few dozens and hundreds. So basically, about 54 of these loci mapped to coding regions in 55 unique genes, so suggesting that we are recovering some signal that could be of interest. And what was also interesting was that we recovered a much higher number of loci for tolerance rather than for the other two phenotypes, really pointing to the polygenicity of this trait. So tolerance also had a higher frequency of loci mapping to genes rather than to intergenic regions relative to the other two phenotypes. And it was also interesting that we actually did not recover canonical resistance genes such as ERG11 or CDR1 or 2. And this could be due to the rarity of the SNPs in these genes or to their narrow distribution within the population. So what was also interesting was the clustering of this loci, right? So we found that about 87% of loci that that were associated to susceptibility really mapped to chromosome 5, whereas a total of 77% of tolerant loci mapped to chromosome 3 and also in particular to chromosome 6. So this suggested that these genomic regions might really be important for azole responses, and perhaps this wasn't all that surprising given that azole resistance and aneuploidy has really been associated with some of these chromosomes, and this is a throwback to work from Anna Salnecki and others identifying some of these aneuploids being associated with azole response phenotypes. So you can see these peaks here in the chromosome 5, 3, and 6 for tolerance. So because the chromosome 5 region in particular looked like essentially one broad association peak spanning around 60 kb, and because it also overlapped a dense set of genes, many of them which were quite interesting, including cell wall, membrane, and even ERG11, we asked whether this region actually harbors one or multiple signals. So are the different SNPs still associated once we account for all the SNPs nearby, because this is a very dense region, or not? So to answer this, we ran a conditional and joint analysis, which revealed that this region actually decomposes into several independent signals, not just one lead SNP. So importantly, these signals here, which I've highlighted in green, actually partitioned by phenotype. So for example, we have loci that seem to be associated with both tolerance and heteroresistance, tolerance and susceptibility, tolerance alone, or tolerance and susceptibility, really validating our findings and really illustrating the polygenicity of some of these drug responses. So to summarize some of our findings, susceptibility loci mapped to 11 genes, which had roles in membrane transport, cell integrity, lipid homeostasis. For tolerance, the loci mapped to a much higher number of genes, so 37 in this case. And these were largely involved in stress responses, drug adaptation, lipid, mitochondrial functions, ion homeostasis, protein homeostasis, and genome stability. Whereas for heteroresistance, we uncovered a much fewer number of genes, so only 7 genes. And these seem to have roles related to or predicted to be involved in genome integrity, cell cycle, telomere maintenance, and cytoskeleton organization. So our next step was really to assess the role of these genes in azole responses. So here we leveraged our collections of knockout and overexpression mutants and measured their susceptibility, tolerance, and heteroresistance using the methods that we used initially for our large set of isolates. So we tested about 100 mutants in total, and what I'm showing here are just those mutants that have significant differences in either susceptibility or tolerance. So by MIC, I'm really referring to susceptibility. By SNG, I'm referring to tolerance. But the heat map here shows genes that were identified to contain loci related to susceptibility. So these values here are showing susceptibility and tolerance values of mutants relative to wild type strains. And the ones marked with asterisks are showing the ones that are significantly different from the wild type. So what you can see here in the genes that were associated with susceptibility is that while several of them do affect susceptibility, many more actually affect tolerance. So this was quite surprising for us. Something similar we noted with genes that are important for tolerance or appear to be important for tolerance. Most of them, most of the ones that we tested actually did have phenotypes related to tolerance. So you see changes both for the knockouts or for the overexpression, but several of them also affected susceptibility. Really, this confirms that while the two traits are genetically separable to some extent, there are also important overlaps that are worth investigating further. And it's also important to note that the loci that we are identifying in our GWAS analysis, they come from different genetic backgrounds, while the mutants that we have are in a single genetic background. So there's also this context-dependent note that we should, be aware of. So finally, for genes that were associated with heteroresistance phenotypes, we identified 5 genes that had significantly different heteroresistance levels and actually to quite high extent. And the main trend that emerged from these mutants is that really these genes seem to be involved in genome integrity, cell cycle, and maybe other functions as well. So really, this is a lot of information. There are a lot of mutants and a lot of genes. Like, what are the take-home messages that we're taking from here? So basically, I did a fun exercise to take all the genes that we recovered from our GWAS and generate a word cloud using the current CGD annotation. And you can see several themes emerging. So we see strong signature for cell surface biology, so cell wall, plasma membrane, GPI-linked factors, lipid homeostasis, alongside also words that relate to stress responses as well as nuclear and genome maintenance, indicating that, again, azole responses involve a broad adaptive network and diverse cellular routes that are important to consider and maybe target at the same time, perhaps in combination with azole drugs. So, moving to a slightly different topic, because a significant number of our isolates were aneuploid in our set, we also wanted to zoom in into these a bit. So, what we found was that the majority of them were trisomies. So, most of the aneuploids that we found were trisomies. And in particular, trisomies of chromosome 5 and chromosome 7 seem to be enriched. We're also curious to see how they are distributed in the phylogenetic tree. So to no surprise, we found that these mapped to diverse clades. So really aneuploidy was not restricted to a particular clade. There wasn't a particular clade that was associated with a higher rate of aneuploid cells, aneuploid strains. What was even further interesting was that if we were to split our isolates into 2 different groups, euploids and aneuploids, and then look at their drug responses separately, we find that aneuploid isolates actually show more than twofold higher resistance relative to euploids. And you can see their distribution here, the distribution of isolates according to their MIC50 levels. When we zoom into this further, we actually find that there are no chromosome-specific effects detected in our data set, suggesting that the genetic context probably matters a lot more than the identity of the chromosome itself. So thinking about the take-home messages of my talk today, I actually went back to my conclusion slides from my previous MycoTalk, so almost 5 years ago, where I tried to emphasize that there are multiple routes that cells can take to withstand antifungal treatment. So it feels like the message is the same here. It hasn't changed that much, and this is still my take-home message today, but we've certainly added more wrinkles to the story in that we better understand the mechanistic basis for this phenomenon now. So, for example, while for resistance, we have a lot of evidence pointing to the importance of, altering the drug target or modulating drug efflux to get to resistance. We now understand that to achieve tolerance, you can ion homeostasis, lipid homeostasis, stress responses might be important. While for heteroresistance, it seems that really genome instability might play a big factor. So this also highlights why single target therapies often fail, and why we need combination approaches that address multiple adaptive layers. And of course, potentially some of these adaptive layers and different routes of adaptation might occur at the same time. So we might need to target them at the same time as well. So finally, I want to thank the people who contributed most to this work, especially Kyle Schutz, who was a main driver of this project, who now moved from University of Colorado to his own position. People in the lab, Cécile Gauthier and Corinne Moffret, who did most of the phenotypic and genomic analysis, as well as collaborators at Pasteur. And thank you for your attention. I'm happy to take any questions.
Thank you so much, Jules. To remind the audience that we will be taking questions at the end, and please submit your questions to the Q&A, not to the chat, because we're not monitoring that. My name is Sarah Gaffin. I'm from the University of Pittsburgh, and it's very much my pleasure to introduce our next speaker, Bernhard Hube. Bernie is, of course, essentially a household name in the fungal world. He did his PhD at the University of Göttingen and his postdoc at University of Aberdeen and then the University of Hamburg. In 2000, he moved to the Koch Institute in Berlin, become a research group leader and the head of division. And in 2006, moved to Jena in Germany, which is where now, as professor and chair in both the French Schiller University of Jena and the Leibniz HKI. I think everyone in this audience is probably very familiar with Bernie's beautiful work identifying Candida albicans, virulence components, the SAPs, candidoleucin, and so much more. He has amazed me with discovering the components and what happens in infection, but really the big picture of evolution and how evolution drives commensalism, drives virulence, and how they're balanced. So not just the what, but the why. Every time I see him speak, I come away understanding so much more than I did. I'm not alone. Bernie has a long legacy of incredibly successful trainees. Neil Gao pretends to be one of them. But it's truly an honor and a privilege, and I think the title of this talk is Commensal Virulence School, so we will all learn something today. Bernie.
Okay, thank you very, very much, Sarah, for these kind words. And it's a pleasure for me to speak it with this audience. And yes, I would like to speak about Candida albicans and the commensal virulence school. So let me start with this slide showing the environmental fungi, how many fungi exist in the world. So it's estimated that there are 3 to 5 million fungal species. So it's a huge, huge number. But if you look at the number of pathogenic humans, it's very small number. Only 150 to 400 species are pathogenic for humans. So that's less than 0.01% of all fungi which are potential for humans. So there must be some selection, some specialization for these very few fungi. And they come from 2 different sources. Either they are from the environment, like Aspergillus, Blastomyces, Candida auris, Coccidioides, Cryptococcus, Histoplasma, and many more. And then there are the endogenous fungi. So fungi which are already living with us on our surface or within us. And these include Candida species, Malassezia, anthrophilic, dermatophytes. But in my talk, I would like to entirely focus on Candida albicans. So Candida albicans is a commensal, but it's not always a commensal because they're environmental and animal associated isolates. And we investigate some of these from samples worldwide, which we obtained worldwide. And of note is that Amelia Barber here in Jena found that the environmental isolates found of the genomes are within the known clades. So that's not a big surprise. But then we collaborated with Alex Hübner and Tina Roringer here in Jena and in Leipzig, and they analyzed the genomes from a calculus of a gorilla skull about 150 years ago. And this genome is a bit different from the other clades. And so are Candida albicans genomes from Paleophysis from Mexico, about 1,500 years old. Again, they are a bit different from the other clades. But I would like to stress here is that these data show that humans have been colonized with Candida albicans for a long time. So let's describe the different lifestyles, commensal and pathogenic lifestyles. As a commensal, the fungus is on mucosal surfaces. It's up to 95% associated with humans, so we are all carrier of this fungus. And this is the normal lifestyle of Candida albicans. But as we all know, the fungus can also be a pathogen. I would call it the most common endogenous fungal pathogen. It's an opportunistic pathogen, can cause disease under certain predisposing conditions. As a commensal, in terms of the numbers, the gut is the main reservoir. And here the fungus is within the lumen or within mucus. And I would propose that occasionally there's local contact to epithelial cells. As a pathogen, Candida albicans can cause superficial infections, oral, vaginal, and skin, and it can cause colitis or systemic infection, up to systemic infection, life-threatening and systemic infections. As a commensal, it needs commensal factors. That means growth without damage. That means nutrient acquisition, and it also means competition with the microbiome. As a pathogen, it needs virulent attributes. That includes adhesion, invasion, and damage, growth within tissue, and immune evasion, of course. But I would like to stress that this is common, and this is the exception. Nevertheless, I will start with the pathogenic stage. And Geraldine Butler, in her Nature paper, Geraldine Butler and co-authors found that there are gene families enriched in Candida albicans. This paper found that there are gene families significantly enriched in the more common pathogens, and Candida clade pathogens show expansions of extracellular enzyme families. And these include lipases. And this is my last first author paper 25 years ago, the discovery of the secreted lipase gene family in Candida albicans. And even older is my first paper my first paper 35 years ago. We discovered with Bernhard Hube and Frank Otz the first protease gene, the first subgene. We thought this would be the protease gene, but then it turned out it's also a member of a family, the subgene family. And I would like to stress that some of these genes, like sub1 to 3, are yeast associated, while some are hyphal associated. Sub4 to 6 are hyphal associated. That brings me to the key virulent attribute of Candida albicans, the yeast to hyphal transition. And this occurs under a number of conditions: contact with surfaces, serum, temperature, CO2, and so on. Many triggers which trigger the important yeast to hyphal transition. But I would also like to stress that also regulation on the reverse way, hyphae to yeast conditions, like quorum sensing, and yeasts are required for dissemination. Hyphae, on the other hand, are important for virulence, not only because of the morphology per se, but because it's associated with the virulence program and adaptive prediction, expression of genes required for the next stages of pathogenicity. And I wrote this in 2004, at least one transcriptional program associated with the yeast to hyphal transition. This not only allows immediate adaptation to changing environmental conditions, but also prepares cells for subsequent steps of infection. This includes the expression of sub4 to 6, PRA1, SOT5, ALS3, HWP1, and EC1. Virulence program, adaptive prediction. Let's start with adhesion of Candida albicans to epithelial cells. These are yeast cells in liquid on plastic, and they float around, and they stop floating because they produce hyphae. And hyphae are sticky because hyphal-associated genes include ALS3 and HWP1. And these are the main adhesins of Candida albicans. They are found on the surface of hyphal cells, but not yeast cells. And you can find this not only on plastic, but also in vitro on epithelial cells, but also in patient samples. These sticky hyphae are attached to an oral epithelial cell. So there's adhesion, ALS3, HWP1, virulence program. Then invasion of Candida albicans into epithelial cells. This occurs via two routes, induced endocytosis and active penetration. Induced endocytosis is an entirely host-driven process. And only hyphae can do this because ALS3 is the main invasion. ALS3 binds to E-caterin, and that induces uptake by host cells, induced endocytosis. And then there's active penetration, and this is a fungal-driven process. You can kill the host cells, the fungal can still invade. Again, only hyphae can do this. But if you look underneath the entrance, active penetration, induced endocytosis, and if you look at this one here, more closely you can see the membrane surrounding this invading hyphae. And it's even more clear if you take trans-electron microscopy, you can clearly see this membrane surrounding this hyphae, and we call this the invasion pocket of Candida albicans. Right, adhesion and invasion, ALS3, virulence program. Now damage. Damage is due to EC1, and that is because EC1 encodes a polyprotein containing candidialysin, a pore-forming peptide toxin. This is peptide number 3, and the synthetic peptide number 3 alone can cause epithelial damage. And this is because it's a pore-forming amphipathic peptide with 2 alpha helices termed candidialysin. And it's highly expressed in the invasion pocket. By using an anti-candidialysin antibody, you can see high numbers in the invasion pocket. So from the fungal perspective, what is the advantage of damaging the host during invasion? Now, it certainly is nutrient acquisition. We have recently shown this for zinc. So the fungus has access to zinc in the cytoplasm, and it can grab up zinc via PRA1. PRA1 grabs zinc from the host, delivers the zinc to ZRT1, which is a PRA1 receptor, but also a zinc transporter. And so the fungus can get zinc from the cytoplasm. The same for iron. Iron can be get from ferritin, and the ferritin receptor on the surface of hyphae is again ALS3. And that needs damage. And also the fungus has, with damage, access to proteins, which can be used as a nitrogen source. And now it makes sense why when EC1 is expressed, there's also ALS3 expressed for iron acquisition, PRA1 for zinc acquisition, and sub4 to 6 for proteolysis. Adaptive prediction. Right, how does the host respond to damaging hyphae? Now, at the very early stages, there's membrane stretching, maybe within the first 20 minutes. But after that, there's repair. And repair is including attraction of ALG2 as a repair protein, but also the escort machinery. But there's also blabbing. Blabbing, shedding of blebs with plasma membranes, which are full of candidialysin. So the host is trying to get rid of contaminated membranes. And what is the evidence for this? You don't see this with the EC1 mutant. Right, so there's moderate hyphae-mediated damage, which can be repaired and tolerated. That can be seen as a commensal activity as well. But what happens when the level of damage is overwhelming and they cannot be compensated by repair anymore? In this case, candidialysin triggers the danger response pathway as identified, discovered by David Moyes and Julian Naglick, and Julian now calls it the epithelial activation mechanism because it's quite complicated. And I'm not going into any detail, but I would like to stress that the trigger of this pathway includes the attraction of phagocytes. And now it makes sense why SOT5 is expressed together with EC1, because SOT5 encodes an extracellular GPI-anchored superoxide dismutase, ROS detoxification. So if you cause damage, you might expect phagocytes which produce ROS. Adaptive prediction. Right, now let's look at the commensal stage. As I said, the commensal stage is the common stage. Therefore, the evolutionary pressure is here. And this is where the virulence properties are trained. And I call this the commensal virulence school. So if Candida albicans has been trained in the commensal phase, what has the fungus learned? Now let's first look at lipases. Why secreted lipases? Why an entire family of lipases? To investigate this, Osama in our department has disrupted all 10 lipases. So he produced a LIP1 to 10 lipase mutant, which was, of course, ideal for investigating the role of lipases. And lipases permit growth on fat, so cow butter, olive oil, and so on. And you can see the mutant is hardly growing. It's not growing at all better, but with the wild type it's growing. So secreted lipases are required for growth on high fat as a carbon source. And to investigate the role for colonization, we joined forces with Richard Bennett, and Chapman did these experiments for us. And you can see that during a high-fat diet, the wild type outcompetes the LIP1 to 10 mutant. So you need the lipases for colonization. This is an antibiotic model, but you can see the same results without antibiotics. So with and without antibiotics, the wild type outcompetes the mutant, the LIP1 to 10 mutant, in a high-fat diet, not low-fat. So there's hardly any differences, or not, at least not significant differences in this case. So with other words, secreted lipases are required for colonization on high-fat diet with and without an intact microbiome. But next, the question, which morphology, yeast or hyphae in the commensal phase? Now, the dogma based on standard mice colonization models using antibiotics is yeast is required for colonization and hyphae for pathogenicity. There are many very good papers showing that yeast block mutants are stable colonizers of the gut, forced hyphal formation impairs gut colonization, and experimental evolution selects hyphal impairment during gut colonization, and hyphal-associated genes like SUB6 and HYR1 are detrimental for colonization. However, filaments are prominent in the oral mucosa. Yeast and hyphae are found during gut colonization, and most clinical isolates of Candida albicans can form hyphae. And there's secreted IgA targeting hyphal virulence factors. That suggests that the host must have seen hyphae. So why is this conserved, yeast to hyphal transition, for a fungus which is mostly a commensal? And why is this conserved, producing a toxin as a commensal? Now, to investigate this, we joined again forces with Richard Bennett's team, and Richard found, Shabnam and Shenhua found that in a germ-free mice, but also in antibiotic-treated mice, the EFG1 yeast block mutant can outcompete a hyphal-competent wild type. But if you include bacteria, then it's the opposite. The wild type, the hyphal-competent wild type, outcompetes the EFG1 mutant. And that is the same with the EC1 mutant. So you need Candida lysin for colonization, and because the wild type is outcompeting the EC1 mutant. Right. And the reason for this is shown here. There's direct inhibition of growth and metabolism of bacteria by Candida lysin. So there's reduced growth in the case of Candida Escherichia coli. There's reduced metabolic activity, and there's also reduced glucose consumption. And we found this for E. coli, but also similar data for Enterococcus and Klebsiella. So there's direct inhibition of bacteria by Candida lysin. So there's selection pressure. This explains the selection pressure to keep and produce Candida lysin in the commensal phase because it's an antimicrobial peptide. But the question is also, is Candida lysin unique to Candida albicans? No, it's not, because it's found in related species. And John Richardson et al. Found that it's found in Candida dublinensis and Candida tropicalis. but it's also found in Candida maltosa, so a non-pathogenic Candida species. And they all have the same structure. There are these 8 different peptides, and peptide number 3 is the Candida lysine autolog, and it's directed by lysine arginine site, so very similar. And Tony Gabaldon in Barcelona found out that the ancestor of all these Candida lysins is actually 30 million years ago. So that's far before humans actually existed. And all of these Candida lysin autologs can inhibit bacteria. This includes the ancient Candida lysin shown here. They all inhibit growth of E. coli. We found similar data for Enterococcus and Klebsiella. So with other words, Candida lysin has evolved as an antibacterial toxin. So this would summarize this part. Candida lysin as a commensal factor. There is in polymicrobial environment, Candida lysin can be used against other microbes, and in the case of Candida albicans, in the host niche, in the gut, there's local antibacterial effects. Right. But why is this conserved? Why are hyphae? Why not Candida lysin producing by yeast cells? Now, the explanation which I would give you is that, as I said at the beginning, Candida albicans hyphae in the gut occasionally, transiently invade epithelial cells. And while they do so, they produce Candida lysin and sub-4-6. And this, in turn, induces local inflammation. And local inflammation, in turn, would induce an immune response, including the production of antimicrobial peptides and reactive oxygen species. And these would inhibit bacteria more efficient than Candida lysin alone. And we tested this hypothesis, and we could find that Candida albicans is much more resistant towards antimicrobial peptides like defensins and LF37 as compared to bacteria. The same is true for resistance to reactive oxygen species. Candida albicans is more resistant to H2O2 as compared to E. coli, Klebsiella, and Enterococcus. So Candida albicans is more resistant to human AMPs and oxidative stress. And if you add Candida lysin, low concentrations of Candida lysin, together with low concentrations of LF37, you have a synergistic effect. You can clearly the inhibition or even killing of E. coli in these combined conditions. So Candida lysin can act synergistically with AMPs against bacteria. And that can also be tested using a competition assay. If you use Candida albicans and E. coli and add Candida lysin, then Candida albicans already outcompetes E. coli, but that's far more drastic if you do the same in combination with either H2O2 or LF37, or in combination of both. So E. coli disappears more or less. So Candida albicans outcompetes E. coli in the presence of Candida lysin and or LF37 or oxidative stress. So that would explain the scenario: local inflammation, production of AMPs, ROS, that would support the fungus and it could compete with the microbiome. And you have sub-4-6. And now it makes sense to have sub-4-6 in this scenario because sub-4-6 is known to degrade AMPs. So this would be a counteractivity against the induced AMPs, which of course can act also against the fungus, but I propose this would also occur under higher induction of local inflammation. So if the fungus goes too far, there's loads of AMPs and ROS, and this would inhibit Candida albicans, in particular hyphae as well. And that is supported by other antimicrobial peptides like peptide YY or secretory IgA, which act specifically against the fungal morphology. And now it would make sense for the fungus to exist not only in the hyphae form, but also in the yeast form. Right. I would like to stress also that Candida albicans can also be seen as a commensal factor independent of the microbiota, as published recently by Salome Leib und Gut Landmann together with Ricardo in collaboration with us. And I would like to summarize this as stressing that candilysin is a classical virulence factor, but also an avirulence factor and a commensal factor. It depends on the stage, the dose, and the context. To summarize my talk, in the commensal phase, Candida albicans produces candilysin, it's inhibiting bacteria. Then there's occasionally invasion, which is good for nutrient acquisition for the fungus. There's repair, there's tolerance, but then there's occasionally local and transient inflammation, which would further inhibit bacteria, would give the fungus a competition advantage. And this is learned in the commensal virulence school. This is all required for the commensal phase. So production of hyphae and candilysin is good for the commensal phase. And then under certain conditions, predisposing conditions like the use of antibiotics, this turns into virulence factors. Now there's fungal overgrowth. Now it turns into a virulence program and adaptive prediction. And there's candilysin-mediated necrotic damage. There might be translocation. But there might also be, at a certain stage or early stage, protective immune responses. And this again would switch the scenario back to the commensal phase. So this is co-evolution. The fungus has learned to produce a toxin, and the host has learned to deal with that and to counteract. So in principle, all mysteries solved, as my team has produced this title for me for my 50th birthday, so only recently. But I was just better take this away and focus more on my favorite MPM team shown here. This is the group who did the work. And I would like to thank everybody on this picture and everybody named here for the excellent work. It's so much fun. It's so great to collaborate with you, to work with you. Thank you very, very much. I would like to stress the EC1 team, Tim, Jacob, Hans, Johannes, and Steffi. And I would also like to thank Julian Naglik, my friend and colleague, who collaborated with me on almost all EC1 projects, together with David Moyes and John Richardson. There are loads of recent and ongoing collaborations as shown here. I would like to thank everybody as well for the collaborations. Funding agency, German Research Foundation, BMFDR, European Union Wellcome Trust funded these projects. And I would like finally to thank all my friends and colleagues in the fungal community. It's so much fun to work at this community. Thank you very much.
Bernie, that was fantastic, as was that wonderful talk by Jules. And Sarah and I, Bernie, can you now unshare your slides? Thank you. Yeah, Sarah and I will try and capture the questions which are coming into the Q&A, and please do continue to populate the Q&A box with your questions. Remember not to use the chat, but the Q&A. And we will probably just alternate through these questions and see how far we've got in the time we've available. We've got quite a bit of time for questions. Okay, so let's kick off. And Jules, may I start as a question here? Thank you for your interesting talk. But asking about this work, which is probably mostly done in vitro, but how can you interpret this in vivo? Do these heterogeneous phenotypes occur in patients? And how would you investigate this?
Yeah, so we know from a bunch of other species that these phenotypes are relevant in vivo. Indeed, everything that I've shown today was done in vitro. So for example, heteroresistance and azoles in Candida glabrata was associated with in vivo persistence of infection. In parapsilosis, heteroresistance to echinocandins this time was associated with breakthrough fungal infections. In Cryptococcus neoformans, azole heteroresistance was associated with cryptococcal meningitis. So there's a lot of evidence showing that these phenotypic heterogeneity phenotypes are important. Having said that, we did also show that there is an association between higher tolerant isolates and persistent infection. And we do recover aneuploid isolates from infections that are resistant. And once you passage them in the absence of drug and they lose this resistance, they also lose the aneuploid chromosome, suggesting that there's a link between isolates that are aneuploid and their resistance levels, suggesting that heteroresistance could arise in vivo, although we haven't done a lot of experiments in setups like this to fully demonstrate it.
Thanks very much, Jules. Over to you, Sarah.
So we have a very short, sweet question here. Does Candida have antifungal activity?
Right. That's a very fair question, and we have tested this in fact, and it's not, or fungi are not susceptible against Candida lysans. So we tested this for Candida albicans, we tested this for related species, and that seems to be the membrane, which is resistant. We can also use protoplasts, and there's still no damage. And we used artificial membranes mimicking fungal membranes, and it looks like the fungus is resistant, or the fungal membrane is resistant. That might be due to the specific lipid composition. We have had ergosterol as a potential protective component, but that has not been confirmed. And I would like to stress that that all other membranes which we have tested so far are susceptible, except potentially the phagosomal membrane. So that would explain why you need hypha formation to escape from the phagosome, because you need the physical forces. Because if you have phagocytosis, the fungus immediately produces hyphae within the phagosome and produces Candida lysan, we know that, but this does not damage the phagosomal membrane. This, in addition to the question.
Okay, well, if I take the next one, Jules, is aneuploidy more frequent in heteroresistant isolates, given that their phenotypes are more transient?
We actually don't see this in our isolates. So most of the heteroresistant isolates that we uncovered are actually euploid isolates. In terms of whether they can become aneuploid faster, I think the jury is still out there. We do see that they are more able to become aneuploid in response to drug exposure. So, for example, to fluconazole.
Okay, thank you. Sarah.
So, Bernie, there's a question about to what extent does Candida lysan dysbiosis in the gut related to that? Are there different bacterial susceptibilities, or does it really equal opportunity on all bacterial types?
Yeah, we are working on this as well. We have screened a number of bacteria, and I would say there's no specific effect against pathogenic bacteria. Some seem to be more resistant, and some seem to be more susceptible, but there's no. It's all bacteria, and we are currently also working together with Alan Walker in Aberdeen on the anaerobes, which might be susceptible. Yeah, it's on the way.
Okay, Jules, there's a question here posted by Arya Murthy, who's interested in your GWAS studies and is imagining a study with multiple drugs. And she asked the question, do you think that tolerance GWAS hits would be more familiar across different drugs, I suppose you do with different drugs, than susceptibility GWAS targets? If so, is there any trade-off for the tolerance alleles?
Yeah, I think that's a great question, and I would love to answer this more directly, but I can speculate that maybe tolerance GWAS hits would be more different across different drugs, because tolerance phenotypes in general reflect stress adaptation and damage mitigation pathways that are likely to be less drug specific than the loci that we might recover in response to susceptibility to different drugs. In terms of the trade-offs, I think, so we haven't measured this, but I would assume that they will carry context-specific trade-offs. So for example, if specific stress response pathways are upregulated, or you have altered membrane composition, this might lead to some fitness costs in particular contexts.
Great. Thanks so much.
The question about whether there's an antibacterial effector in the Candida yeast phase that is distinct from Candida lecem.
Sorry, which question is this? Sorry.
Whether there's an alternative antibacterial effector
Alternative.
expressed in the yeast phase.
Right. Okay, got it. Yes, antibacterial activities. There might be. I'm not aware of yeast cells having antibacterial activities. And the question also included whether there's a bit of candidalism produced by yeast cells. No, there's nothing. EC1 is entirely hyphal expressed, and it's one of the highly expressed genes by hyphae at all. And there was, before it was discovered that EC1 encodes for a toxin, EC1 was frequently used as a marker for hyphal formation. So people were using EC1 expression and say if EC1 is expressed, then there must be hyphae. But I'm not aware of antimicrobial activities of yeast cells. There might be.
I may elaborate on that a little bit. Is there reason to think there would be? Or is it that the Candida yeast really are living in sort of anonymity among all the bacteria that is around them?
But our focus was on the question of why hyphal formation is conserved. And that is probably because, I mean, yeast cells can also colonize, but they are outcompeted in the presence of bacteria.
Bernie, can I just ask for a sort of postscript on that? EC1, I think that's the original acronym for extent of cell elongation. So some intermediate levels of expression from pseudohyphae. And how do you see pseudohyphae fitting into the school of virulence, the school of commensalism? Is it also just simply an intermediate?
Good question. Has anybody found pseudohyphae in the gut?
I think if you look at any clinical sample, you'll find some cells which are different, and I think probably you'd have to say yes. Yeah,
okay. But I mean, it's also difficult to analyze it because you don't have the detailed mutants for preventing pseudohyphae formation. But yes, of course, it could be something which is in between.
Okay. And Jules, another question which is at that sort of lab clinical interface. And the question is, since current CLSI and UCAS reference methodologies are not reliably detecting tolerance and heteroresistance in Candida isolates, for example, in the MIC test, what tests would you recommend to detect these isolates in clinical labs? Will the detection have to be relied on whole genome analysis, or could some phenotypic methods be used that could be feasible in clinical labs?
Yeah, I think that's a great point. Detection based on whole genome analysis is not there yet because we don't have specific genetic markers that we can use to pinpoint these phenomena. However, people have known about tolerance, but have been calling it something else for a very long time, and they have been observing this phenotype actually in CLSI and UCAS methods. So these phenotypes are apparent in current assays. For a lot of them, you might just need to wait a bit longer to observe them. So we can see tolerance in liquid susceptibility assays, and we can measure it. We can see heteroresistance on solid plates, so either UCAS, test strips. Of course, we cannot measure it, and we would need a more detailed analysis, such as a PAP assay. But even for PAP assays, we can certainly reduce the amount of work and plates needed by using a single concentration that's above the MIC. So I don't think we need something extremely expensive or extremely sophisticated to notice these phenotypes. I think we might just need longer incubation time and a trained eye to detect them.
Okay, thanks very much. Sarah.
So, Bernie, there's a question about what triggers the hyphal transition in the commensal state.
In the commensal state, yeah.
Fungal factors.
Very fair question. I would say in the commensal stage, at least the gut, it's N-acetylglucosamine. So that's the most obvious one because it's a cell wall component of bacteria. And there are also papers about describing that there is even increased N-acetylglucosamine after antibiotic treatment because the cell wall components are floating around then. So this, for me, would be the most obvious trigger in the gut.
And outside that.
Okay. So Jules, that's a question from Lucy Z. Do you know what is the relationship between tolerance and resistance, if any? For tolerance, how do cells within the same clonal population exhibit different drug responses?
Hi, Lucy. I think that's a fantastic question. So I don't know how to answer either of these questions. So we're still trying to understand the relationship between tolerance and heteroresistance, especially because oftentimes we see them both occurring in isolates. And I think in terms of how cells from the same clonal population exhibit different drug responses, you are an author of the first single cell assay analysis that has asked this. So we know that some cells will continue to grow, while others will be more focused on exhibiting stress responses. But we really don't know what happens upstream and really like what decides what cell does what. And I think that's a very important question that we're definitely trying to understand in the lab right now. Thanks.
Okay, and maybe I could just add another question on the back of that, and it's about heteroresistance in a different organism. So it's from Beatrice Aachen, and have you had a chance to determine heteroresistance in clinical isolates of Cryptococcus neoformans, and if they're associated with poor clinical outcomes?
Yeah, we haven't done that, but others have. And indeed, heteroresistance is present in Cryptococcus neoformans cells in response to azole drugs, and the presence of heteroresistance has been associated with cryptococcal meningitis. So we think that phenomenon is important in that species as well.
Okay, thank you. Sarah.
So, Bernie, the question, you might have partially answered this, but is there evidence for candidylation expression in non-free-forming Candida yeast, which I think you said there are, and what are the explanations for their adaptive strategies?
Yeah. That's, of course, a very good question. There are autologs in other species, but are they expressed? And under which conditions are they expressed? That's a very important question. And in the paper from John Richardson, he investigated, or he and his team investigated Candida lysans from Candida topicalis and Candida duplinensis. And the synthetic peptides are even more toxic to epithelial cells than Candida albicans, Candida lysans. So they're highly toxic, but the genes are not expressed. So that's I would propose this might be an evidence that they are not used, these autologs are not used for pathogenicity, but for use for something else. Could be contact with bacteria, like in the ancient scenario and in the non-pathogenic Candida species. And we have tried very, very hard to find expression patterns. We have created reporters for a number of these species, but so far we couldn't find any conditions where these genes are expressed. So in our hands, they are silent. But they must, I mean, they are conserved, they must be expressed somehow. So we haven't found the trigger yet.
Yeah. Yeah, I was wondering, you mentioned Candida maltosa, Bernie.
Yes.
That's an interesting organism, isn't it? I assumed it might probably be something to do with nutrition from what you were saying.
Possible, yeah.
Okay. So, Jules, I've got a question from Hung Ji Sai. If there's no specific aneuploid karyotypes associated with tolerance or heteroresistance phenotypes, could GWAS analyses reveal more common loci after excluding aneuploid isolates from your collection? So get rid of them first and repeat the analysis. Yeah.
Yeah, we actually already did this because we had a similar question from a reviewer, and we're actually recovering about 90% of the same loci. So it doesn't make a big difference. And I think that's because the aneuploids that we have are not a substantial part of our data set. So they're about 12%. And of course, this might influence some of the details of our analysis, but both analysis with and without the aneuploids give us the same core of stringently significant loci.
Okay, thank you.
We have a question from Jezebel. A clinician, are we close to separating commensalism from causal pathogens in the presence, in this case, in vaginal symptoms for Candida albicans versus non-albicans species?
Yeah, that's a very fair question, especially for vagina infection. Yeah, thank you very much, Jack, for this question. I don't have a clear answer for this, but I would say there's not a clear cut because between commensalism and pathogenicity. So as I try to stress in my summary, I guess it's sort of question of threshold. At a certain threshold, it might turn into pathogenicity, it might turn into symptoms, and if the host response is too high, it's also detrimental for the fungus in the gut. But in the vaginal scenario, it might be that that's not a clear cut. That's for sure. I would like to stress in this context that also in the vaginal scenario, it is also to be considered that the virulence function, which I mentioned, that Candida albicans is recognized by the immune system, and the immune system can induce a protective immune response that does not work in the vagina. In this case, there's more and more attraction of neutrophils, and here Candida albicans is really a virulence factor. So this is whenever it is expressed and is causing inflammation at a certain level. Yeah, difficult to say. Is it now? That's not if there are neutrophils attract, then it's not commensal growth anymore. Question of threshold.
Okay, Jules, this is another one. People are obviously interested in this sort of clinical laboratory interface of the work. How do you discriminate in a patient between heteroresistance and a clinical strain population that consists of resistant and wild-type susceptible cells? If resistance emerges, it takes time to outcompete or outgrow the wild-type cells. So I can imagine this looks like heteroresistance at first glance.
Yeah, I think that's an excellent point, and I wish I would have a good answer for this. You're entirely correct that if you, let's say, recover an isolate from a clinical setup, you do a PAP assay, you might recover a small percent of resistant colonies. And just using that PAP assay, you won't know if you have a clonal resistant subset of population that's just emerging and hasn't yet taken over the population, or the formation of rare heteroresistant cells. So you would need a follow-up analysis for this. You would need to, so really the distinguishing trait between heteroresistance and true resistance is its transience. So if you take some of those colonies that you get on the PAP assay and passage them without drug, the loss of the resistance phenotype over time will indicate heteroresistance. However, this of course can vary from isolate to isolate depending on the genetic background. So some will lose the resistance faster than others. So I don't have a definitive answer for how long you might need to passage them or things like that. But yeah, you're entirely correct that we cannot discriminate between the two just using a PAP assay.
Yeah.
I'm gonna take Tara's privilege and ask a question that's not on the list there. With respect to the lipases, Bernie, if impact of particularly in terms of high-fat diet, is that straight to the gut, or are there different impacts of this lipase cluster depending on the anatomical location of the infection? So oral versus vaginal versus gut.
Whether there are differences depending on the site, yes. This is also stressed in the paper from Ricardo and Salome Leibengut-Landmann. Here, Candida albicans is a commensal factor without the microbiome being involved. So that's different.
I was thinking about this though, in particular, because of the different fats and fat content, dietary lipid content that might be present in the oral cavity versus the gut, for example.
Difficult to say. Don't know.
Okay.
Okay, I think we're probably coming to the end because our seminar speakers were so on time. We've had to be able to have a whole bunch of questions, but there is one still to ask you, Jules, and it's from Charlotte Smith, and she's asking you. First of all, she like everybody is saying, great talk. Very interested in the result that there was no trailing growth, which is another terminology for what you've been describing, in Candida glabrata. Was this expected? Are there any reasons for this?
Yeah, so the slide that I showed really is a snapshot of one single study in one particular country. So the distribution of these phenotypes tends to differ from hospital to hospital. So Candida glabrata also exhibits tolerance. So that wasn't the definitive picture of those isolate. It was just to illustrate the prevalence of this phenomenon. Yeah, and in general, it's harder to detect tolerance for drugs that are cidal rather than static. So I think there's a much smaller window for tolerance in those for those drugs than for azoles. But yeah, great point.
Okay, and Sarah, back to you because another question's popped in
Okay.
right
Well,
at the
I could go
bottom.
back to an earlier one, which really asked both speakers to maybe address a couple of key takeaway points that a clinician might want to know from this talk that you gave today.
Yeah, so two take-home points for the clinical perspective. Jules, you want to go first?
Sure. I guess I can repeat what I said earlier. I think some of the susceptibility assays that are done in the clinic could be incubated for longer for some of these phenomena to become apparent. And I think improved training and knowledge of the existence of these phenomena will help clinicians actually be able to detect them in those settings.
Yeah, I could say that candidialysin clearly is involved in inducing inflammation, and that depending on the mucosal site, it might make sense to block candidialysin to stop production of inflammation, in particular during VVC, because VVC is overreaction of the immune response, while on other sides, the immune response can be protective. So we have tried using our nanobodies to neutralize candidialysin, and it works. It works in vitro. We haven't done that in vivo, but it works, and it's similar, I would say, similar successful as the PRA1 story from Duncan. So it might be that PRA1 and candidialysin are key targets to prevent VVC, and that you can block PRA1 and EC1, or not EC1, candidialysin, to prevent inflammation during VVC.
Okay. And Sarah, do you want to take there's a last question, this from Joe
Oh, sorry,
Hyman.
there it is. Yeah. There's a question of whether growth and cancer expression are triggered by particular bacteria.
Right. Not to my knowledge. We haven't found this.
Okay. I saw a question from Joe Hyman, but I can't see it anymore. I don't know what's happened to it.
I answered it on chat.
Yeah, you did. Okay. That's what so you've
Yes.
typed the answer.
Yes.
Okay. Well, I think we will probably start to wrap up then. So, I mean, I just want to thank both Jules and Bernie for outstanding talks. Really interesting. We had a huge audience, and if you notice, we had over 220 plus people during the talks, which is wonderful. I do also want to thank my wonderful co-chair, Sarah Gaffin. It's also a pleasure to work with you, Sarah. And to remind everybody that on the 26th of February is the next MycoTalks, and we will be entertained by George Deep and Tamara Doring on that occasion. So do look out for that and register for that. And thanks everybody for all their really interesting and probing questions, which also lended a lot to the excitement of the event. So thank you very much, everybody, and have a wonderful day.
And I would also like to quickly thank the folks behind the scenes who are making this work as seamlessly as it is, especially Jenny.
Okay, well,
stay warm, everyone.
Yeah, try to stay warm. Bye for now.
Thank you.
Thank you.
Goodbye.